Welcome back, students. Let's talk about lipid-soluble hormones, specifically their mechanism of action. Before we do that, though, let's review just a little bit. Let's review water-soluble hormones. So if we look at a water-soluble hormone, remember they do not need a carrier. They have no trouble going through the blood. So here comes our hormone. It's this little red dot. And look, it doesn't just fit anywhere. It fits into a specific spot with a very specific shape on the receptor. And it fits like a lock and a key. So once the hormone binds to the receptor, remember it's going to act through this G protein. It's going to activate a G protein. The G protein in turn is going to activate adenylate, or in this case, adenyl cyclase. And what we're going to do is we're going to take ATP, chop off two phosphates, and create a cyclic AMP. Well, as long as this enzyme is working, we're going to continue to make cyclic AMPs. And so this can make a lot of cyclic AMP. Well, cyclic AMP really only has one job, and that is to activate protein kinases. And when we activate protein kinases, they in turn have only one job. And what they do is they activate or inactivate other enzymes. And so we get this cascade. So let's now talk about lipid-soluble hormones. So remember lipid-soluble hormones. Here's some examples. You probably recognize these: testosterone, estrogen, cortisol, also the leukotrienes and prostaglandins, even though we're not going to really talk about them. And then remember there's one amino acid-based hormone that is lipid-soluble, and that's T3, T4, or thyroxine. But they're all going to work the same way. They're all going to work by direct gene activation. So remember what's going to happen is we have to have a transport protein. Lipid-soluble hormones will not dissolve in the water of the blood. So they travel through the blood attached to this protein. When they reach their target cell, they're going to detach from the protein and leave the bloodstream. If you look here at number 1, there is no extracellular receptor because a lipid-soluble hormone has no trouble diffusing across the bilipid layer. So it enters the cell with ease. What it's going to do then is it's going to find its receptor. Once it finds its receptor, we're going to create a hormone receptor complex, and together those are going to interact with the DNA. Let's look at it in a little more detail. So, they won't dissolve in water, must have a carrier. They do not need an extracellular receptor. The receptor's on the inside because the hormone can enter the cell with ease. They're going to bind to the receptor, and we're going to create this hormone receptor complex, which is going to go to the DNA and cause the DNA to begin to produce messenger RNA. We're going to get transcription. That messenger RNA then will enter the cytoplasm, and it will bind to ribosomes, and we'll create new proteins. We're going to do translation. And it's those new proteins which are going to create the effects produced by this hormone. So if we look at it, we can see it here. So let's look at it a little closer. This is a steroid hormone. Remember, steroid hormones are lipid soluble. It could be others. It could be a leukotriene, or it could be a prostaglandin. They all work exactly the same way. Now, this hormone has already been released from its carrier, and it's about to cross the cell membrane. And look, it just zooms right across. It has no trouble. It also zooms into the nucleus without any problem. And what's going to happen is it's going to hook up with a receptor. The receptors are floating around in the cell, and they're escorted by these little chaperones. They're moved around, and they're going to finally hook up. The chaperones will be released, and we're going to get this receptor hormone complex. Together, these two are going to go to the DNA, and they're going to bind to the DNA. They don't just bind anywhere, though. They bind to a very, very specific place associated with a very, very specific gene, and they're going to turn that gene on. Well, when you turn a gene on, what we're going to get is transcription. So we're going to make a copy of that gene, but not DNA. We're going to make a copy as messenger RNA. And if you remember from general biology, messenger RNA is sort of a recipe or a list of instructions about the order to assemble a protein. Which order do the amino acids need to go in? Remember, we have codons and anticodons. And what's going to happen is we're going to take this messenger RNA, it's going to go to the ribosomes, and we're going to decode it. And as we decode it, we're going to build a brand new protein. And it's the new protein that's going to produce the effects that this hormone has. Let's look at that real quick. So here's our lipid-soluble hormone. Remember, it must have a carrier. Now it's already detached from the carrier. We can see the two of them here. The carrier is the green part, the hormone is the red part as it travels through the blood. But when they get to the cell, it immediately detaches and zooms across the membrane. There's our receptor. Now, this receptor happens to be out in the cytoplasm, but they hook up, they're going to go to the DNA, they're going to interact with that DNA, we're going to get transcription, and we're going to make a messenger RNA. A messenger RNA is going to go to the ribosome, we're going to decode it, and what we're going to do is we're going to build a protein. We're going to assemble the amino acids in the correct order according to the instructions until we get a brand new protein. So that's how lipid-soluble hormones work. Well, I think you probably already realize that not all cells are going to react to all hormones. It has to have a receptor. And any cell with a receptor is called a target cell. So target cells, the ones with receptors, are the only ones that are going to react to a hormone. Hormones go everywhere. They circulate to all the tissues in your body, but they don't affect those tissues. They only activate cells that have those receptors. And the receptor binds to the hormone. It can be an extracellular receptor if we're talking about a water soluble, or an intracellular receptor if we're talking about a lipid soluble, but it binds to the hormone. They have to bind like a lock and a key. Once they bind, then we get the effects. Well, let's look at these target cells. There are some hormones that only have a few target cells in the whole body. If you look at the hormone ACTH, it stands for adrenocorticotropin hormone. It comes from the pituitary. And the only place in your whole body that has receptors for this hormone is on the adrenal cortex. So even though the hormone circulates with blood and goes everywhere to your heart, your lungs, your liver, they ignore it. The only cells that have receptors, and so the only cells that respond to this are on the adrenal cortex. So ACTH has a very narrow effect. On the other hand, if we look at another hormone, thyroxine, remember this is T3, T4, there are receptors for T3, T4 on basically all the cells in your body. So that means when the hormone is released into the blood goes everywhere, but it's not just one part of your body that reacts to it. Almost all parts of your body react to it. And so thyroxine has very widespread effects. Now, when we bind to the target, we bind to the receptors, we get an activation, and we get some sort of a response. Well, the response, the size of the response that we get, depends on more than one thing. It actually depends on three things. One thing is how much of the hormone is there. The more hormone we have, the bigger the effect. Another one is how many receptors are actually on the target cell. I think it makes sense that if there are only 2 receptors on a target cell compared to another target cell that has 100 or 1,000 receptors, we're going to get a bigger effect. The more receptors there are, the bigger the effect. But there's one other thing that's also important, and that is how well do the hormone and the receptors fit together. Remember, it's like a lock and a key. If they don't fit together exactly the way they're supposed to, it's not going to work. And that's called affinity. So the affinity is how well they fit together. And the higher the affinity, the higher the fit, the bigger the response we're going to see. So 3 things. 1, amount of hormone. 2, the number of receptors. And 3, the affinity of the receptors. Another thing is, when we look at hormones, we usually talk about hormones one by one by one, but that's not the way hormones are in the body. If I were to take a blood sample of a person and look for hormones, I'd see a bunch of them. I get what's called a hormone profile. So all those hormones are in there at the same time. And some of those hormones will interact with each other. Now, some don't. But if they do interact, they can interact in different ways. There are 3 ways in which they interact. One is called permissiveness, another one is called synergism, and then the last one is antagonism. These are 3 different ways in which hormones interact. So let's talk about them. Permissiveness is kind of like giving permission. And what this is one hormone cannot exert its effect without another hormone being present. So that other hormone is giving it permission. Synergism is when you have a combined effect of 2 hormones that's bigger than you expect, bigger than the sum of the 2 individual effects. And then finally, antagonism is when you have one hormone doing exactly the opposite of another hormone. So 3 different interactions. One other thing about hormones is they're very, very, very powerful. You do not want too much of a hormone, just like you do not want too little of a hormone. And so, in order to keep them from being too much or too little, they have to be regulated, just like everything else in the body. And there are 3 ways in which they're regulated. There's humoral regulation, neural regulation, and hormonal regulation. If you look at this graphic, you can kind of see what's going on. Let's take them one at a time. So if we look at humoral regulation, the word humor is an old word meaning fluids. And so humoral regulation refers to something that's in your body fluids, something that's in your blood that's not a hormone. That's what's controlling the levels or the response of this hormone. It usually is something like an ion or a nutrient. So, an example of this would be a hormone that we looked at in in part 1 of A&P, and that hormone, remember, was PTH, and another one, calcitonin. Both of these are related to the amount of blood calcium ions. So remember, what happens is we're monitoring the number of blood calcium ions, and if those ions, the level begins to fall, the parathyroid hormone, PTH, will be released. And then PTH is going to cause the calcium levels to come back up again. And so That's humoral regulation because calcium is not a hormone. It's either a nutrient or an ion. Well, it's an ion. So we have this calcium in the blood. As calcium levels fall, the parathyroid glands, which we'll talk about a little bit later, detect it. And they, in turn, are going to release parathyroid hormone, PTH, and PTH will bring the calcium back up. But who's in control of the release of PTH? Who's in control is the calcium ions. So these glands are humorally regulated because an ion in the blood determines whether or not this hormone will be released. Let's look at neural regulation. So neural refers to the nervous system. And remember, the nervous system works by neurons. It works by nerve fibers. And so what's going to happen here is the nervous system is in control of the release of the hormone. There's going to be a nerve going to a gland, and that nerve, the action potentials, the release of the neurotransmitters, is going to tell the gland to secrete. So if you look at this gland, this is the adrenal medulla. It releases the hormone epinephrine and norepinephrine, but it does so in response to stimulation from the nervous system. So the spinal cord sends out an action potential. It goes down and we get the release of a neurotransmitter. And then the gland responds to that neurotransmitter by releasing epi and norepi, the catecholamines. So who's in control here? Who controls whether or not this hormone is released? The nervous system does. So this is neural regulation. And finally, there's one more way in which it's regulated, and that's hormonal. In hormonal regulation, one hormone controls another hormone. One endocrine gland controls another endocrine gland. If you look at that, it looks like this. So look, we have three sets of glands here. We have the hypothalamus, we have the pituitary, and then we have the glands below, the thyroid, adrenal, and gonad. Well, the way the hormones come out of or are released from the thyroid, the adrenal, and the gonad is because they're told to by the hormones from the pituitary. The pituitary releases those hormones because they're told to by the hormones of the hypothalamus. So the hypothalamus releases hormones that control the pituitary. The pituitary releases other hormones that control the thyroid, the adrenal, and the gonad. And then those glands release their other hormones. So who's in control of the thyroid gland? It's actually the hypothalamus. The hypothalamus releases a hormone, and then the pituitary releases a hormone. So the thyroid gland is hormonally regulated. So three different ways in which they're regulated. So look at this one, and you can see sort of the generic hormonal control. So there's our hypothalamus up at the top, and then there's the pituitary, and then there's our endocrine gland, whatever it is, and finally there's the target cells. So we start with RH, that's our releasing hormone. That goes to the pituitary, which releases its own hormone, which is hormone 1. That goes to the endocrine organ, which then releases its hormone, which is hormone 2. So who's in control of that endocrine organ? Who tells it to secrete? And who tells it to secrete is a hormone. So it's hormonally regulated. So 3 different ways in which endocrine glands are regulated.